Okay, guys. Now we go to the cell cycle topic. So we're gonna start with this objective. At the end of this topic, the student, you must know how to describe the phases of the cell cycle, and then you can differentiate between mitosis and meiosis, and lastly, you can describe how the cell cycle is regulated. Okay, what is cell cycle? So the cell cycle is a sequence of events. So it's an event, okay, from A to B to C and until to Z. Okay, so these events leads to the cell division. The first one is cell division, and second one is replication, meaning that the replication is can be considered and can be known as a duplication. Okay, so in prokaryotes, the cell cycle is through the process of binary fission, meaning that from one cell it divided into two cells. Okay, division in half. So DNA replication and segregation occur simultaneously at the same time. Okay, so in unicellular organism, so it happened as a for reproduction. Okay, and for multicellular organism, it's also for reproduction and functioning as a whole organism, which replacing the dead cells. Okay. Okay, now we go to the subtopic of it, which is it contains the chromosomes, the mitotic spindle, and the kinetochore. Okay, now there's a few terms that you have to know that you need to know, which is the chromatids, the centromere, the sister chromatids, and the kinetochore. Okay, first we go to the chromatids. So the chromatid is one of two strands that form when a chromosome replicates. Okay, meaning that it's one of the double-stranded, so one of the two strand which happen during the replication. Okay, that one is called chromatids. Okay, and the replicated chromosome has an X shape at metaphase. Okay, so this is X shape. You can find it only at the metaphase of the mitosis. All right, then for centromere, so the centromere is the part of the replicated chromosome that is connected to the spindle fiber. As you can see the images down there, so there's a kinetochore in pink color, and then it's connected with spindle fibers. Okay, all right. So then we go to the sister chromatid. So what the sister chromatid is? So both replicated chromosome is also known as the sister chromatid. As you can see that the blue color, you can see the blue color of the chromatids, and on your left and on your right there's the maternal chromosome, which is a pink or red in color. Okay, so the sister chromatid is the X-shaped one. Okay, and when it goes together with the blue and the red, it called homologous pair, or also known as the non-sister chromatids. Okay, so the kinetochore is a protein structure that forms on a chromatid during cell division, and this allows it to attach to the spindle fiber on the chromosome. So that's called a kinetochore. Okay, now there's a three types of microtubules that involve in the cells during for the mitosis and meiosis. Okay, so if you still remember that the previous lecture, I have told you about the types of cytoskeleton, which consists of the microtubules, the intermediate filaments, and the microfilaments. Okay, so this is microtubules. Okay, so microtubules in during this process, there's a three types involved. The first one is the ester, or we also known as astral microtubule. Okay, so this microtubule will grow towards membrane, meaning that it outward of the cells to the membrane. Okay, and this is for the cell stability. Okay, the first one. So the second one is a polar microtubule. So this microtubule extends towards the opposite pole, meaning that from right to the left. Okay, and then this is for the cell elongation and also the stability. That's for polar microtubule. So polar means from left to right, or from north to the south. Okay, that's why it's called polar. Okay, and then the third one is the kinetochore microtubules. So this microtubule attached to the kinetochore at the centromere of each sister chromatid. So this is for the chromatid movement and separation. So when you, as you can see on your right, the kinetochore microtubules is in dark green. Okay, in the dark green, that's connected to the centromere of the chromatids. Okay, for the polar microtubule, as you can see here, it's in a light green light green color. Okay, for the polar is from the left to the right. Okay, and for the astral also the light green color, but it's towards the membrane. Okay, so all these thing is called microtubules, or we call it a mitotic spindle. Okay, so the mitotic spindle is a cytoskeletal structure that organizes and separates chromosomes. So all of this is called the mitotic spindles. Okay, and it's made up of the microtubules, and these microtubules form to organize the centers of organelles. Okay, now we go to the interphase, the mitosis, and the cytokinesis. Okay, the phases of cell cycles can be divided into two general phases. So the phases are, the first one is interphases, and second one is the mitotic phase. Okay, so in interphase phase, um, it consists of the G1 phase, the S phase, and G2 phase. Okay, so interphase is the stage during which the cells prepares, grows, and accumulates nutrients needed for mitosis, and also duplicates the DNA. So this is the event that happened in the interphase. Okay, so during interphase also, the progression from one phase to another is regulated by phase-specific expression of pro-proliferative cyclins, okay, and their cyclin-dependent kinases. Okay, so we will look into detail this thing, the pro-proliferative cyclins and the CDK, okay, and also the anti-proliferative tumor suppression. So all these things are important to inhibit the cell cycles. Okay, we will go into detail after this. Okay, that one is for interphase. So the second one is the mitotic phase. Okay, so in mitotic phase, the cell splits itself into two distinct cells known as daughter cells, and the final phase is cytokinesis. So bear in mind that the mitotic phase consists of two different phases or two events, which is the nuclear division. That the first one. The second one is cytoplasmic division. So in nuclear division, there's a mitosis and meiosis. For cytoplasmic division, there's also called a cytokinesis. All right. Okay. So before we go to the G1 phase or interphase, there's another phase that you should know, which is, we call it a G0 phase. Okay, or also known as a quiescent phase. Okay, this is the phase where the cells are in the resting phase. Okay, the cells has left the cell cycle and stopped dividing. Okay, and it's also referred to as a post-mitotic phase. Okay, for this cell which is in this phase, it's carrying out their normal metabolic functions. Okay, but it's not replicating. Okay, therefore it is not in the cell cycle. All right. Okay, so normally the cells that can be considered in this phase, for example, as neuron, and also the fully differentiated cells like muscle cells. Okay, meaning that the cells cannot be divided anymore, and it only can maintain and do their own metabolic functions. Okay, all right. Then, so when a G0 cell is signaled to replicate, it moves into the G1 phase. This is for another case, whereby the G0 phase, whereby the cells was signaled to replicate, and then it will move into the G1 phase. Okay. However, the cell with DNA damage will arrest at G1. Okay, at G1 checkpoint, or at G1 phase. Okay, and many cells do not enter the G node, and normally the cells continue to divide throughout an organism life, like epithelial cells lining in the mouth, lying in the mouth and gut. Okay. Okay, G1 phase. Okay, so G1 phase is the first phase in the interphase. Okay, so this phase also called a growth phase. Okay, normally cells respond to the external signals such as growth factors by manufacturing of the cytoplasmic nuclear contents and protein needs for daughter cells. Okay, so normally the cells that will go into G1 phase receive the external signals. Okay, external signals from outside of the cells. Okay, so they will prepare the cells to become to become a two daughter cells. Okay, for example, they induce sufficient production of activated complexes, so meaning that this complexes is sufficient, then it's ready for to go for the synthesis phase or S phase. Okay, so we will go into detail for the cyclin D and the CDK4 and 6 complexes after this. Okay, all right. So if cell is under unfavorable conditions, meaning that the cells in stress, the cells have a DNA damage, or the cells has a lack of nutrients, so the cells will enter a resting state known as a G naught phase, as I mentioned before. Okay, so it's called a cell cycle arrest. Okay, okay, if the cells pass the restriction of the G point or point of G1, so it will go or move to the DNA replication phase, or also known as a S phase. Okay, once the cells has passed the G1 phases, so it will go to the S phases. So in S phase, the cell starts the DNA replication starts. Okay, so when the phase completes, so all the chromosomes have been replicated. At the end of this phase, each chromosome will have two sister chromatids. Okay, and a pair of sister chromatids are joined together at a DNA region known as centromere. So all the sister chromatids will assemble at and in the middle of it, or at the center at the centromere. Okay, and also during this phase, the amount of DNA in the cell is doubles. Okay, but the ploidy of the cell remains unchanged. Okay, so the ploidy, if you remember that there's a haploid and diploid. So the diploidy means the meaning that also known as the diploidy is the number of complete sets of chromosomes for each cells, okay, in each tissue, and it depends on different organism. Okay, all right. So in this phase, the synthesis is completed as soon as possible. Okay, because in this phase, the DNA is very sensitive because the base pair is exposed to the external factors like drugs and mutagens. If you still remember the previous topic about the DNA replication, about the nucleic acid. So once they has received the external factors, it will open the wine or the wing, open the double stranded, and it will happen to commence the DNA replication. That's why it will complete as soon as possible to avoid these external factors to make the cells or to make the DNA mutant. Okay, after the DNA has been replicated, they will go to the G2 phase. Okay, so G2 phase is a gap phase. Okay, which happens during the gap between the DNA synthesis. Okay, the DNA synthesis and the mitosis. Okay, so in between, meaning that from between S phase and M phase. Okay, so the G2 checkpoint mechanism, it controls to ensure that the cells is ready to enter the M phase or the mitosis phase and divides. Okay, and these checkpoints restrict progression to M phase if error are found in the duplicated chromosome. Okay, so they will check they will check whether the protein synthesis is enough for it to go to the mitosis phase. Okay, if there's a significant error, it may trigger apoptosis. Okay, now M phase. Okay, so mitosis takes place only in eukaryotes. Okay, remember this is only happen in eukaryotes. Okay, the chromosomes in the nucleus of the cell will be divided into two identical nucleus. Okay, and then in M phase there's two different things: the mitosis and also the cytokinesis. Right, so these two are also known as the mitotic phase in the cell cycle. All right. So the mitosis is the biological sum of four phases. Okay, the four phases meaning the prophase, the metaphase, the anaphase, and telophase. All right. So in these phases, it distributes all the replicated chromosome that happened in previous phases during the DNA replication phases or the S phases. All right. So again, still remember. And then when the mitosis is complete, the genetically equivalent chromosome exists at the opposing poles of the cells. Okay. At the end of these phases, there's a two different the equivalent chromosome exists at two different sides of the cells. Okay, and this stage is followed by the cytokinesis. Okay, so the cytokinesis will start in the middle of the anaphase phase. Okay, so from anaphase to telophase. So this is where the cytokinesis happen. Okay, cytokinesis divides the mother cells into two daughter cells, and in cytokinesis, the cells, then the cells, nuclei, the cytoplasm, organelles, and all, and the cell membrane also is divided into two equal shares. So it will at the end have a two identical cells. Okay, now, so this is the details of the mitosis process. Okay, so normally prior to mitosis, each chromosome will make an exact duplicate of itself. Okay, and it become thickened. Okay, and then in early prophase, most of the centrioles will start to move apart into two sides. Okay, into two poles. Okay, and the nuclear membrane starts to disintegrate. Okay, start to disappear. All right. Okay, in the late prophase, the centromere, the doubles, the what you call it, the chromosome is in the middle. Okay, and the nuclear membrane almost disappeared, and all both the ester or the centrioles are now at the opposite poles. Okay, and then during metaphase, the double chromosome will line up at the middle of the cells. Okay, and after that, at the early anaphase, the chromosome is divided into two, will split into half. Okay, whereby half will go to one pole and the other half go to another pole. Okay, that's for the early anaphase. For late anaphase, all the half of the chromosome will reach to this to the both pole. Okay, and if you can see that in the figure, there's a dent, or there's a curve whereby it shows the what you call it, the divided the cytokinesis happen, or cytokinesis begins in this phase. Okay, and then at the telophase, the nuclear membranes will start to form. I mean, the nuclear membranes start to form, and then the the cells are ready to be divided into two. And at the end of this process, there's two cells with the same structures produced. There's two daughter cells produced in this process after the mitosis happened. Okay, now we go to the meiosis. Okay, meiosis is different with the mitosis. Okay, so meiosis in human, it will make the diploid cells into the haploid cells, meaning that from two end into one end. Okay, and in human, the haploid cells make the meiosis that make the meiosis. For example, the sperm and eggs. Okay, so when the sperms, which is N, and egg in another one end joined together or fertilized, it will become the two haploid sets of chromosome. Okay, so it will produce a new genome. Okay, so this meiosis is from two n divided into n. Okay, so and then in human, there's a sperms and eggs. So the sperm is one n, the egg is one n, and fertilized together, and it will become the haploid sets. Okay, and it will produce a new genome. All right. In this meiosis phase, there are two different phase involved. It's divided into two different phases. So the meiosis one and the meiosis two. All right. So the meiosis one is whereby the homolog pairs separate. Okay. For the meiosis two, the sister chromatids separate. Okay. So in each round of division, cells go through four stages. The same thing. The same like mitosis. The prophase, the metaphase, the anaphase, and the telophase. Okay. Since cell division occur twice during meiosis, so one starting cell can produce four gametes. So eggs or sperm. Okay, so as I said before, also consists of the prophase, metaphase, anaphase, and telophase. Okay, so the prophase, during the prophase one, the homologous chromosome pair up and exchange fragments. in the middle of the cells. Okay, as you can see, the first one figure, there's a centriole start to move towards start to move to other poles. Okay, and then both homologous chromosome will try to pair up, and there's a event called an exchange or crossing over the pairs. Okay, and then after that, in this starting cells is the cells in diploid. Okay, meaning that it's 2n. So 2n will have 4 chromosomes. Okay, and the metaphase 1, the homolog pairs will line up at the middle of the cells. Okay, whereby the centrioles is already at their poles. Okay, in anaphase 1, the homologous started to separate to the opposite ends. Okay, but the sister chromatid stays together. Still remember the sister chromatid? The sister chromatid at X1, okay, the X shape, okay. Homologous chromosome is double X shape. Okay, remember there's a 2X shape for homologous chromosome. For the sister chromatid is X-shaped chromosome. Okay, so in this anaphase 1, the sister chromatids will stay together. Okay, but the homologous will be separated. So this is anaphase 1. Okay, at the end of that, the telophase 1, the newly forming cells are haploid, which means that n will be that from 2n it will become 1n. Okay, each chromosome has 2 non-identical sister chromatids at the end of this phase. Okay, now is the phase of meiosis 2. Okay, so meiosis 2 start with the prophase 2. Okay, the start cells are dihaploid. You still remember before this, it's n, not 2n. Okay, it's n. Okay, so n will start to the starting it will the chromosome will condense in the middle of it. Okay, so the membrane nucleus will start to disappear. Okay, and then metaphase 2, in metaphase 2 phase, the chromosome will line up in the middle. Okay, this is called the chromosome, sister chromatid. Okay, the sister chromatid. Okay, yeah, will line up in the middle of it, in the middle of the cells. Okay, and in anaphase 2, the sister chromatid will separate into 2, and it will go to the opposite ends of the cells. Okay, at the end of the process, which is the telophase 2, the newly forming gametes are formed. Okay, this is haploid. Okay, haploid. Okay, and then it will have from one cells will become 2 cells. So from here you can see there's a 2 cells, so it will become 4 cells at the end of it. All right, so this table I have prepared a table for you. It's easier for you to study, so I think you could look at it on your own. Okay, whereby I tried to gather all the information which show the differences between meiosis 1 and meiosis 2 in terms of the according to their phase. Okay, the prophase, the prometaphase, the metaphase, and anaphase, and outcome. What is the outcome of the for each event? Okay, now we go to the cell cycle checkpoints. Okay, this is the important thing, the important part of the topic. Okay, so if you still remember the figure of the cell cycle before this, there's a 3 different checkpoint. Okay, so the G1 checkpoint, the G2 checkpoint, and the metaphase checkpoint. So these those 3 checkpoint is very important. So checkpoint is a stage for the cells to examine. Okay, whether it's favorable, whether the cell is favored to pass another phase to before it goes to another phase. Okay, meaning that from prior to go to a second phase, so it must check whether the cells is competent or not, the cells is favored or not. Okay, so for G1 checkpoint, in this checkpoint they determine the appropriate growth condition to check whether it has sufficient nutrients, how about the cell size, and then the presence of the protector. So in this checkpoint, the level of the DNA damage is will be assessed. Okay, that one is for G1 checkpoint. In G2 checkpoint, in this checkpoint, they determine the state of the pre-mitotic cells to see whether it's suitable, the suitable cell size required for the successful division is can be met or not. Okay. Okay, so in this checkpoint also it can identify a replication fault, okay, whether there's the changes or DNA sequence or not. Okay, and then the last checkpoint is meta-fifth checkpoint. This is to ensure the proper spindle assembly and the correct attachment to the centromeres. So this point is important to prevent the non-disjunction events. All right, so this is the images or the figures about the checkpoint. Okay, that's the G1 checkpoint, G2 checkpoint, and the meta-fifth checkpoint. So in each checkpoint, there's the things that need to be assessed. All right.